Hello everybody, and many thanks for joining in for this spotlight session focusing on new mechanistic insight to ER-alpha biology and endocrine therapy. As you probably know, breast cancer is the most common cancer in women and represents a large fraction of newly diagnosed cancer overall. Over 70% of breast cancer are considered hormone dependent, since the primary driver of proliferation is the activation of the nuclear receptor transcription factor estrogen receptor alpha. All adjuvant treatments are designed to interfere with estrogen receptor activation. New first-line therapies target protein downstream of the same pathway, for example CDK4 and CDK6. Overall understanding of hormone-dependent breast cancer initiation and evolution requires a profound molecular understanding of estrogen receptor function. We already understand a great deal about estrogen receptor biology. Upon estrogen binding, the activated receptor binds DNA at region called enhancer, which controls gene expression by forming long-distance loops to gene promoters. More recently, several groups have uncovered how ER-alpha binding on the DNA has the potential to scar the genome in addition to its transcriptional stimulation capacity. For example, Periazami and colleagues have discovered that estrogen stimulation can induce double-strand DNA breaks on the genome, as shown here by H2X and POL2 damage. In abstract 466, Nicola Chephagen and colleagues explored the connection between DNA damage and paradoxical estrogen-induced cell death. It has previously been noted that cells that acquire resistance to endocrine therapy generally end up with higher level of estrogen receptor. Additionally, one potential collateral effect of endocrine therapy resistance is that estrogen re-exposure can trigger now cell death rather than cell proliferation. This observation underlies current clinical trials that aim at using estrogen exposure to control tumor burden in relapsing patients. Nicole used an ER-alpha positive cell line, in this case HCC1428, and its endocrine therapy resistant counterpart, the 1428 LTD, or alternatively an ER-alpha positive T47D cell line engineered to overexpress estrogen receptor upon exogenous stimulation. In both cases, exposure to estradiol induced cell death rather than cell proliferation, but only in cells with an ER-alpha overexpression. Again, leveraging this ER-alpha positive overexpressing cell, they could show that estrogen exposure led to massive increase in double-strand DNA damage, here identified by gamma H2X staining, and modified histone deposed at site of double-strand break. Additionally, they show that ER-alpha overexpressing cell can also accumulate large number of DNA-RNA hybrid foci, commonly referred as R loops. Using RNase H1, an enzyme which can effectively remove these hybrids, they could show that the DNA damage induced by estrogen requires indeed R-loop formation. R-loop are problematic for cell, and removal of these structures require PARP activity. The author therefore tested if inhibition of PARP activity via a PARP inhibitor, olaparib in this case, could promote further cell death. They tested this hypothesis in two patient-derived xenograft models from endocrine therapy-resistant patients, and could observe a strong synergistic effect, suggesting a novel use for PARP inhibitor outside of the regular or common setting of germline BRCA1 and BRCA2 mutations. This data raise few interesting questions. How do we define overexpression of estrogen receptor in patients? Or do other inhibitors of DNA damage response, such as ATM or DNA PK at check1 and check2, synergize with estrogen therapy as well? Or is this effect specific to PARP inhibitor? The paradoxical effect of estrogen in certain models raised the question about heterogeneity. Besides genetic heterogeneity, we now understand how even cell lines present a significant deal of phenotypic heterogeneity. Recent advance in the field has leveraged single-cell RNA-seq profiling to investigate this issue. In its most used iteration, single-cell RNA-seq is performed by capturing individual cells in oil bubbles, along with reagent and barcodes. Bioinformatic analysis then is essential to visualize these large matrices of data, in which thousands of cells are described by thousands of genes. Itomomori and colleagues have leveraged this technology to profile two novel patient-derived xenografts, SC31, which represent a classical primary ER-alpha positive cancer, which grow in the presence of estrogen and struggle in its absence. On the other hand, GS3 has the reverse phenotype, in which estrogen supplementation effectively curbs tumor proliferation. Histological profiling showed that xenograft had extensive degree of heterogeneity, as shown here by ER-alpha, PR, HER2, and Ki67 staining. The author profiled the estrogen response using single-cell RNA-seq to show that in both instances, cell activate the classical transcriptional response cascade associated with ER-alpha activity, as shown here, for example, by increased expression of the progesterone receptor, a classical ER-alpha target gene. However, estrogen increased the number of mitotically active cells only in SC31, but had the opposite effect in GS3, where the number of cells in G1 did increase in response to hormonal stimulation. By subdividing the cell based on estrogen receptor expression, the author suggests that the effect induced by estrogen can occur both in ER-alpha positive and ER-alpha negative cells. The author proposed that this would require some sort of interaction, since ER-alpha negative cells should not be capable of sensing estrogen changes. In an exploratory analysis, the author found that the expression of interleukin-24 is triggered uniquely in GS3 model. They put forward the hypothesis that interleukin-24 can act as a tumor suppressor gene, and this could partly explain the lower proliferation rates observed in SG3. However, there are only two estrogen-suppressing PDSs available to researchers at the moment, GS3 and WIM16. These data clearly indicate that there should be an additional amount of research to develop PDX that are also estrogen suppressing and can be studied further to corroborate this data. Going back to estrogen receptor and its potential to scar the genome, a potential mechanism behind this was put forward a few years ago. Following the observation that the deaminizing enzyme ApoBec3B form a complex with ER-alpha and is required for transcriptional activation. ApoBec3B is part of the ApoBec family, which were initially characterized for their antiviral activity, but took the stage more recently as the potential driver of one of the most pervasive mutational signatures in cancer. Mutational signatures are identified by algorithm based on the scar they leave in the genome during the lifetime of an individual. For example, ApoBec is thought to underlie what is commonly referred as signature 2 or signature 13 in Cosmic database. The reason behind the presence of this signature in breast cancer genome is currently under heavy investigation from any group. Antonio Marra and colleagues wanted to explore the association between the ApoBec signature and clinical features of estrogen receptor positive breast cancer, especially the response to endocrine therapy. Extracting this signature from the genome is tricky and is generally done using whole genome sequencing data, since signature calling requires a large number of passenger mutations. The author wanted to test the idea that this signature could be extracted by a much sparser dataset, such as targeted sequencing. To test this, they downsample larger public datasets and test the correlation between whole exome sequencing, for example, and targeted sequencing, leading to very good result and good overlaps. Next, they leverage the massive impact dataset, which contained 4,595 consecutive breast cancer, to understand what processes have shaped the mutational profile in these large cohorts. By splitting patients in the archetypal hormone-dependent, HER2-driven, and triple-negative breast cancer, they show that ApoBec processes are particularly prominent in hormone-dependent cancer, where they are even more noticeable in the metastatic setting. This increase is, however, not driven, apparently, by increase at single patient level, since they disappear when the author analyzes matched sample, suggesting that ApoBec-driven patient might have an innate poor prognosis. The author also show an interesting association between the signature and driver genes, most notably for PIK3CA, where the patient with active ApoBec signature appear to accumulate more PIK3CA mutation than expected. Finally, by stratifying patients based on ApoBec mutagenesis, they show that patients with strong ApoBec signature have shorter response to AI. While this is just an association, this data raise a question about the potential of leveraging the dependence on ApoBec mutagenesis to possibly prevent the onset of resistant mechanism and tumor heterogeneity. In the last poster, we go back to revisit a relatively accepted assumption. Hormone-dependent breast cancer do not express only estrogen receptor, but also other nuclear receptors, including the androgen receptor. Since this receptor can compete for gene regulation, the field has hypothesized a long time ago that these competitive events could be exploited for treatment. Recent data suggested, for example, that androgen receptor is a tumor suppressor in ER-alpha positive cancers. This would imply that using selective androgen receptor modulators might be a bad idea. However, Li Sichuan Wei and colleagues present evidence that enzalutamide does not actually interfere only with androgen receptor. They do so by knocking out androgen receptor and showing that in classical ER-alpha positive model, these are still inhibited by enzalutamide. On the other hand, removing the estrogen receptor does interfere with the enzalutamide effect, questioning the prevalent model of action. Saturation binding curve using radioactive enzalutamide also show that these can bind directly to the estrogen receptor and compete with estrogen, reminding us of a classical mechanism of action, the one of tamoxifen. This data therefore suggests that the mechanism of action of enzalutamide in estrogen receptor positive breast cancer needs to be further investigated. With this, on the behalf of all the poster presenters, I would like to thank you for your attention and invite you to ask questions to the author. Thank you very much.